major breakthrough in Huntington's disease, let's talk about it. Firstly, I'd recommend you go and have a read of this article because they explain it probably better than I'm going to do here. In this video, I'm going to talk about what Huntington's disease is, and then I'm going to talk about AMT-130, the new therapy for Huntington's disease that's making all the headlines. So, Huntington's disease, this is a genetic disease, meaning it's passed down through families. The way our genes work is we have 2 copies of every gene, one from each parent. And if you have Huntington's disease, you have 1 mutation in your gene. That means that you have a 50-50 chance of passing that bad copy down to your offspring. So your kids have a 50-50 chance of getting Huntington's disease. There's something interesting in Huntington's disease called anticipation, meaning that when the disease is passed down through the generations, the age of onset of symptoms gets earlier and earlier, and the severity of symptoms gets worse as well. So Huntington's disease, we're thinking about a specific gene and a specific protein. So we're thinking about the Huntington gene, which makes the Huntington protein. This is a polyglutamine disorder, and we will get onto that, what that means a little bit later on. But first of all, let's go back to our basics, our analogies, and I like to use my cooking analogy to explain genetics. I've done a longer video on this in the past, so go and have a search for that. But essentially it boils down to this. All of our DNA is a giant cookery book. This cookery book contains the instructions for every single protein we'll ever need to make. These proteins are our final dishes. Within our cookery book, we have specific pages, recipes, and that's what a gene is. A gene is a chunk of our DNA that provides the instructions for making one specific protein. So here we're thinking of our recipe, our gene being the Huntington gene, and our final dish is the Huntington protein. But anyone who's done any cooking knows that you don't go to the supermarket with your whole cookery book when you want to make a meal. What's the point? You make a shopping list, and this is what's called RNA, specifically messenger RNA. And this is a temporary messenger molecule that we use as a shopping list to go and tell us what ingredients, our ingredients are amino acids, we need to assemble together to make our protein. Now glutamine, if we go back to it, is an amino acid. And this is an amino acid made by the combination of a C, an A, and a G in our genetic code. In the Huntington gene, we have an area called the polyglutamine tract. And what this is a whole bunch of CAGs next to each other. So we have CAG, 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 35 times we have that. And what causes Huntington's disease is when we have more than 35 CAGs next to each other. 36 plus causes Huntington's disease. We're incorporating extra glutamines into our amino acids, into our protein, sorry. And where this anticipation that I mentioned earlier comes from is as follows. The whole reason that it gets worse is this. Every time it's passed down a generation, this chain of CAGs gets longer. This is specifically in cases of the disease. So this chain of CAGs keeps growing and growing, which is why we see earlier onset of symptoms and more severe symptoms. But what exactly does this all mean genetically? So we know that we're making this Huntington protein. Let's use another analogy. All proteins have a function. Every protein does something, and they're incredibly important. And in order to do something, a protein has to fold into a very specific shape. And the shape that it folds into is determined by the ingredients, the amino acids that are inside it. So this key is our protein, and its function is to unlock this lock, which it can only do if it's this shape. Now what happens if in the middle of this key I insert a whole long strand? This is where our polyglutamine expansion is happening. The key is no longer going to be usable. It's no longer going to be able to unlock this lock, and it's going to fold into the incorrect shape, and it's going to form clumps inside brain cells, and that's going to cause them to die. So that's what's happening in Huntington's disease. Now that we've got that out of the way, let's talk about AMT 130. And there are some scary words involved here: virus, DNA, RNA, brains. It is quite scary to think about, but please just bear with me. So let's start off. What actually is this treatment? How does it work? Well, this treatment is performed, it's a one-off treatment by injection into the brain. This is neurosurgery performed over 8 to 12 hours, so it's quite intensive. What are they injecting into the brain? They are injecting a virus, and stay with me, please. This is a completely harmless virus. Viruses are quite often used in genetic therapies as a sort of transport mechanism. We can put DNA inside a virus and then transport it safely into where it's supposed to go, and then it can start its work. So this virus contains DNA for a new gene, and this gene produces something called a microRNA. So we inject this virus into our brains, and then this allows our brain cells to start producing the product from this new gene, this microRNA. So what does the microRNA do? Well, let's think back to Huntington's. We have our DNA, which is faulty. We need to make a copy, our mRNA shopping list, and this shopping list is going to go off and tell us to make that harmful protein. What this new microRNA does is that it binds to our messenger RNA, our harmful shopping list, and it removes it. I'm not going to go into the mechanisms of how it removes it, but it stops us writing that. It stops us sending off that shopping list to make the harmful protein. So we're stopping the production of the protein. It's gene silencing. So we're no longer producing that harmful Huntington protein. What does this look like? Well, the findings of this trial are unbelievable. A 75% slowing of disease progression. So where someone without this treatment would get worse over the course of 1 year, someone taking this treatment would be at the same point after 4 years. And that is absolutely massive. And not only is this really, really good news for the Huntington's disease community, but also for the wider gene therapy community as a whole. Thinking about other different diseases that this therapeutic approach could be employed for, because this is sort of proof of principle that this technology works. So it's really exciting times. I hope that this video made sense. If it didn't, please do go and read this BBC News article. Like I said, they've explained it very well, far better than I can. Perhaps that's because they're being paid for it and I'm sat in my bedroom doing it. Who knows? Anyway, thank you for watching. Bye-bye.